Okay, so the next part of our story is something called pyruvate oxidation. And this is oftentimes treated as kind of a minor, unimportant step in between glycolysis and the next major phase of the cycle, which is called the citric acid cycle. But it actually is pretty important as far as what's going on. But in order to understand it, you have to have some understanding of where all of these things are happening inside of the cell. And so if we look at a cell, let's imagine this is just some generic animal cell, bacterial cell, or not bacterial, but some eukaryotic cell. You've got your cell membrane on the outside, you have got your nucleus up here, and then you've got your mitochondria over here. And your mitochondria has got a membrane on the inside, and the membrane is folded upon itself. It makes all these little fingers, right? So you got all these little fingers that are coming out, and this organelle is mitochondria. That's where the majority of cellular respiration is going to happen, that is going to make the most energy, okay? So if we talk about glycolysis, the thing that we just saw, glycolysis is happening out here in the cytoplasm. And if you remember back to that video, I said how every single organism on the planet does at least this part of respiration. And that's because even if you're talking about a bacterium, which doesn't have a nucleus, doesn't have a mitochondria, bacteria can still do glycolysis because that can just happen out in the cytoplasm. So that's what they do. Well, all of the rest of the phases happen inside of the mitochondria. And so pyruvate oxidation is this intermediate step where we're moving from the cytoplasm into the mitochondria. So we're going to go from out here inside of the cytoplasm to the inner part of the mitochondria. And the inner part of the mitochondria is called the matrix. M-A, just like the movie. M-A-T-R-I-X, the matrix. And that's all the way to the inside of the mitochondria. And that's where the citric acid cycle is going to happen. So as we're moving from glycolysis to the citric acid cycle, and we're transitioning from the cytoplasm to the matrix, that's when pyruvate oxidation is going to happen. So we're going to start with our molecule of pyruvate. So we got 1, 2, 3 carbons. And here's our molecule of pyruvate, 3 carbons. Well, we're going to drop one of those carbons, and we're going to make a molecule of something called acetyl. Acetyl is just another sugar, except this time it has 2 carbons. And when we lose a carbon, every time that you see a carbon being lost, it's going to be lost as a molecule of CO2. And so that molecule of CO2 is going to leave. But think about what just happened. We had a molecule of CO2 leaves. That means we just broke a bond between this carbon number 2 and this carbon number 3. Right here, we broke that bond. Well, when we were doing glycolysis over here, we talked about how whenever you break this bond, in this case, it was an exergonic reaction, so we used it to create 4 molecules of ATP. Well, here, it's another reaction. It's an exergonic reaction. It's another one where we broke a bond. But in this case, we're going to pull off the real meat of what it is we want out of these sugars. And that is high energy electrons. All of these carbons have got high energy electrons in their orbitals. And those high energy electrons are way more valuable to us than these little one-off reactions where we get ATP. And so when this molecule of CO2 leaves, it leaves, that carbon leaves, but now those electrons that used to be within this covalent bond are now going to become available for us to potentially siphon off and take. And we're going to take them, we're going to capture it, and we're going to take it over to the subsequent stages of cellular respiration. But we can't just take the electron as is. You can't just pick up an electron and take it somewhere. It's super energetic, it's really small, it's actually extremely dangerous when it's an electron all by itself. It's something called a free radical. And so we're gonna have a little babysitter molecule, a little carrier that is gonna bind to that high energy electron and take it to the place where we can actually use it. And that molecule is called NAD. And when it has the electron on it, it becomes NADH. Okay? So NADH is our product. NAD was our reactant. And so that means that it's going to bind to that electron, bond to a random proton that's hanging around, and now we're going to get NADH. And so that's a super valuable little molecule to have, is that NADH. And now we're down here at acetyl. Well, acetyl is the sugar that we need for the next step, but it can't just go right into the mitochondria. Remember that the mitochondria is an organelle, which means it's surrounded by a membrane. And remember that membranes are semi-permeable, which means that they don't let just anything inside. And so we need a boarding pass. And in this case, our boarding pass, let's go like this, I guess. Our boarding pass is something called CoA. And so we've got CoA over here. CoA is going to bind to this molecule of acetyl, and it's going to produce acetyl-CoA. Acetyl-CoA is accepted by the membrane of the mitochondria. And so after it turns into acetyl-CoA, it can go through the membrane. It's actually going to go through two of them, of the mitochondria. So here's our mitochondria. It can go through both membranes, and it can arrive down here in the inner compartment, which again is that thing we mentioned before, which is the matrix. Now that it is in the matrix, the CoA can leave because we don't actually need that to do any sort of chemistry. We just needed it to allow us to get into the mitochondria. So the CoA is going to leave, and we're back over here at acetyl once again. And now we can start the next step, which is the citric acid cycle.